Hi everyone, this is chapter 14 on infection, infectious diseases, and epidemiology. So we're going to start out with talking about symbiotic relationships that exist between microbes and their host. So the word symbiosis means to live together. And we have symbiotic relationships with countless microorganisms that live on and in our body. There are 4 different types of symbiosis that we discuss: mutualism, commensalism, amensalism, and parasitism. And so just a brief look at what these mean. So when we're talking about this, we look at the two organisms that are in the relationship, and does the relationship benefit, have no effect, or harm one or both of the participants in that relationship. So that's what we're looking at here. So mutualism, just like you would imagine, it's mutual for both of them. All right. So for instance, you have bacteria in your colon, and that colon is supposed to be there, and it is getting a free place to live and food, and in exchange for that, it's producing things like vitamin K for you. Commensalism, one organism benefits, the other one's not benefiting, but it's not harmed. And an example of this are the mites that live on human hair follicles. They don't hurt you, and it certainly is benefiting the mite, but it doesn't do anything for you. But it doesn't hurt you. So amensalism is where one organism is harmed, but the other one does not benefit, nor is it harmed. And the example of this are fungus secreting an antibiotic that inhibits the nearby bacteria. And then finally, parasitism, where one organism benefits and the other is definitely harmed. So tuberculosis bacteria in humans, and lots of different types of worms that we'll be talking about, helminths, later in the semester, that are considered to be parasites. Here is another example. These are termites, and termites, they have a special bacterium that lives in their gut that actually digests cellulose so that they can eat wood. So they're in a mutualistic relationship where both of them are benefiting. So your microbiome, you probably heard about this because there's an awful lot of discussions about the human microbiome and the good bacteria and all that sort of stuff. Your gut bacteria, all of that conversation is very current. So the microbiome is organisms that colonize either the surface of your body and don't cause any disease. They are also called your normal microbiota, the normal flora, your indigenous microbiota. And there's two different types. There are the ones that are there all the time. We call those the residents. And then there's the ones that are there just for a brief period of time. They're called transient. So showing you some examples of normal microbiota here. The little purple or pink things are the microbes. So a little more about your resident microbiota. These are part of the normal microbiota that live on your body, in your body throughout your lifetime, and they're mostly commensal. So I was showing you some examples here you can take a look at in your upper respiratory tract. And that's the other thing that's kind of interesting about the microbiota. They have a specific place that they live in your body, and they're not supposed to live anyplace else. So you have a different microbiota profile, if you will, in your mouth than you do in your throat, than you do in your stomach, than you do in your small intestine compared to your large intestine. They all have their own specialized area where they live. And so they're showing you some examples here with the upper respiratory tract, because the same thing's going on there, right? And so it's giving you the names of the different microbes you would expect to live there, and then some notes about them. Here in the upper digestive tract versus the lower digestive tract. And you can see by looking at the genus for each of those that you have some that are the same, but you have some that are definitely different. And here in the urinary and reproductive tracts, you have bacteria that are supposed to be there. They live there all the time, and they have a purpose. And then finally, on the surface of the skin and in the eyes, the microbacteria that you would expect to find in those areas. The transients, these remain in the body for a short time. They are found in the same regions, of course, as your resident microbiota, but they can't persist in the body. They're in competition with other microbes that are there. They're often eliminated by your own immune system, and they can cause chemical or physical changes in the body. And we definitely know that. We talk about that with changes that we see in people's gut bacteria. So if you're interested in that, there's a lot of literature out there, a lot of reading to do about gut bacteria and how some very small changes can cause some very dramatic physical changes, good and bad, in the way your body functions by changing the profile of your gut bacteria. So, how do we acquire our microbiome? So, human development and animal development happens in a womb that's free of microorganisms. There shouldn't be anything in the womb during development. So, the microbiome begins during the actual birthing process. So, much of the resident microbiota that you have are established during the first few months of life. They did this really interesting experiment with women, moms, and the difference between the microbiota in babies that were born naturally or vaginally and babies that were born via C-section. And there was a dramatic difference in the microbiome that they had in their bodies, even just immediately after birth. And so they started doing this process where they took uh, towels that were put into the vaginal solutions that moms produce during childbirth, and rubbing those on babies that were delivered by C-section. And they developed the same microbiome as babies that were delivered vaginally. So really interesting studies that have been done about how important that process actually is. Opportunistic pathogens, these are ones that are normal microbiota that can cause disease in certain circumstances. For example, if they get to a place they're not supposed to be, like you have certain microbiota that normally live in your digestive tract, but if those get into your lungs somehow or another, then they can cause an infection. This can also be an issue during immune suppression. So if somebody is on immunosuppressant drugs because maybe they've had an organ transplant, or because maybe they're on chemotherapy, so their immune system is dampened down, then bacteria that normally wouldn't cause problems can create an issue. If you have changes in the normal microbiota, so if there are some genetic changes that can happen in those, and then stressful conditions, because that unbalances the very delicate balance between things that can make you sick and things that keep you from being sick. Most pathogens cannot survive for long outside of their host. We talk about this idea of a reservoir. So reservoirs are a site where pathogens are maintained as a source of infection. And there's three different types of reservoirs. There are animal reservoirs, human carriers, and non-living reservoirs. So animal reservoirs, so we talk about zoonoses. These are diseases that naturally spread from an animal host to humans. We acquire zoonosis through various different routes. It can be from direct contact with the animal itself, or the waste from that animal, by eating animals that are infected, and through blood-sucking arthropods, i.e., typically mosquitoes, but there's others that they bite and draw blood on an infected animal, and then they bite you and I, and unfortunately pass that on to us. Humans are usually the dead-end host for zoonotic pathogens, and they are difficult to eradicate. So these are always coming up. They're fascinating, scary, but fascinating. There is an entire branch of the CDC that does nothing but study emerging diseases, and almost always they're coming out of an animal reservoir. So here's some common zoonosis that you will come up against, I'm sure, many times. Tapeworms, flukes or fasciola, malaria, toxoplasmosis, ringworm. Okay, all those kinds of things. Remember that ringworm is not a worm, it is a fungus, and we're going to talk about it later this semester. Here's some bacterial diseases that are common zoonosis: anthrax, bubonic plague, Lyme disease, salmonellosis, some viral diseases, including rabies, hantavirus, yellow fever. Another reservoir is human carriers. So asymptomatic infected individuals can be infective to others. We all know that. We've all heard that you may be carrying something and you're not showing any symptoms, but you can unfortunately pass it on to others. So some individuals eventually develop the illness, while surprisingly enough, some individuals never do. And so that's always the story about Typhoid Mary. So if you've ever heard of Typhoid Mary, she was a cook and she was spreading the disease Typhus salmonelli, which is the causative agent of salmonella, onto the families that she was cooking for. She didn't exhibit the illness, but she was a carrier for that, so she never got sick. Healthy carriers may have defense systems that help to protect them, so they may have a slightly different immune system. That is something that they've been looking at specifically with HIV/AIDS, and they looked at it with COVID too, where they had individuals that had the viral count in their body, yet they never got sick. And what is different about their immune system? And so that's been an area of interesting research as well. And then finally, non-living reservoirs, soil, water, and food can be reservoirs of infection as well. The presence of microorganisms is often due to contamination by fecal material or urine. So how about invasion and establishment of microbes in hosts? How does an infection happen? Well, first you have to be exposed to it, and that is through the process of contamination. So this means that there is the presence of microbes in or on the body. Infection means that an organism has evaded the body's external defenses, i.e. your skin and mucous membrane, multiplies and becomes established in the body. One of my very favorite microbiologists passed on about 15 years ago, and I had the pleasure of working with him for a number of years. And one of the things he used to always say is to be a good microbe, you had to get in and you had to stay in. So meaning good meaning if you wanted to make someone sick. And that's very true. You have to get past the defense systems, and then you have to get within a place that you can multiply as a microbe, and then you have to establish and stay. Portals of entry are sites where pathogens enter the body. There's three major pathways that this happens. One is the skin, another is your mucous membranes, and the third is the placenta. There is also entry via the parenteral route, which circumvents the usual portals. So here's looking at routes of entry for invading microorganisms. So things like your ear and broken skin and insect bites, conjunctiva of your eye, your nose, your mouth. Notice that a whole lot of these are up around your head. Okay. And then, of course, the placenta and reproductive organs. So let's talk about these in a little more detail. The skin. The outermost layer of your skin is dead. So the outermost layer of your epidermis is dead. It's called the stratum corneum, and it acts as a barrier to pathogens. Some pathogens can enter through openings or cuts. So even if you get a little micro abrasion and you look at it a couple of days later, let's say you got a little teeny tiny cut and it's all puffed up and red, that means it got through that corneum layer. The epidermis of your skin is only about the thickness of a paper towel. So that's a good paper towel, like a Bounty paper towel, but a paper towel nonetheless. And that doesn't take much, even a scratch that doesn't bleed, to actually introduce microbes underneath of that epidermal layer. Others can enter by burrowing into or digesting those outer layers of your skin. So again, showing you a cross section of skin and all of the different layers that we have. Thin skin has 4 layers of the epidermis. Thick skin, which is what's on the palms of your hands and the soles of your feet, has 5 layers. And then underneath of that, you have the dermal layer. And then underneath of that, you have the subcutaneous layer or the fat layer. And it's showing you that all of those layers there, even though 2 of them, the epidermis and the dermis, are true skin, and then the one below that's just a fat layer, all of those are protecting the tissues underneath from entry by microbes. Your mucous membranes, these are going to line body cavities that are open to the outside world. So they provide a moist, warm environment that unfortunately microbes actually really like. So your respiratory tract is the most common site of entry. Entry is through the nose, the mouth, or through your eyes. The gastrointestinal tract may be a route of entry as well. It is hard for most microbes to survive the acidic pH of your skin, of your stomach, but there are some that can tolerate that. And then finally, the placenta. This typically forms an effective barrier to pathogens. The pathogens can cross the placenta and infect the fetus. It can cause spontaneous abortions, birth defects, and premature births. So here's some examples of pathogens that are able to cross the placenta. A protozoan one is Toxoplasma gondii. It causes a condition called toxoplasmosis. And so you can see the effect that it can have on the embryo or the fetus. This is the one they caution you about if you're pregnant, why you should not handle dirty cat litter. So you shouldn't be cleaning cat boxes. If you look on the side of a cat litter box, it specifically says that. So cats can carry this and not show any signs of it whatsoever, but it can be in the cat feces that they transmit, and an individual who is pregnant can actually end up with this condition because of the aerosolization of that protozoan. Then some bacteria and DNA viruses, RNA viruses that are all interesting to look at and understand the effect if they were to cross the placenta. The parenteral route. This is not a true portal entry. This means that the portal of entry has been circumvented. So this is like a cut or something like that. It's a different way than the ways I was just talking about. Pathogens are deposited directly into the tissues beneath the skin or the mucous membrane. Let's talk about how do the microbes stay? How do they stick or adhese in an infection? So adhesion is the process by which the microorganism attaches itself to your healthy cells. So this is required to establish colonies that are successful within a host. So again, you got to get in, you got to stay in, and this is the staying in part. They use adhesion factors, including specialized structures and attachment molecules. So it's just showing you here a microbe and how it binds to the host cell surface so that it can stick around and not get washed away or phagocytosed. The attachment proteins that help in this process are found on viruses and many bacteria. Viral and bacterial ligands, a ligand's just a nice way of saying a substance, are going to bind to the host cell's receptors, and that interaction can determine the host cell's specificity. The changing or blocking of those ligands or its receptor can prevent the infection, which is one of the areas that they look at preventative pharmaceuticals. The inability to make attachment proteins or adhesions renders the microorganism avirulent, so it can't make you sick. And some bacterial pathogens attach to each other to form a fascinating thing called a biofilm. So dental plaque is a really good example of a biofilm. It is a whole colony of different kinds of bacteria that are hanging out together. They're attached to each other, and then they attach to your teeth, right? And so that is what a biofilm is. It's of special concern, as I think I might have mentioned in a lab already, but it's a special concern on medical instrumentation. So that's one of the reasons we use the autoclave. A little about the nature of infectious disease. Infection is the invasion of the host by a pathogen. And what results is a disease if the infecting pathogen alters the normal body function. Disease is also referred to as morbidity. So you may have heard of the morbidity and mortality report that doctors often have to submit. And that means how many people got sick and how many people died. So morbidity is the how many people got sick part. So the difference between a sign, a symptom, and a syndrome. So a symptom is subjective. They're things that the patient tells you. Now, when you're dealing with animals, your patient can't really tell you those things. But in a human, it would be things like my stomach hurts or I feel achy. There's nothing you can measure that with. There's not an instrument that measures stomach out at pain, right? Or an instrument that measures achiness. Signs are objective manifestations of disease, things that we can observe or measure. So like your temperature or your blood pressure. Okay, those are all things that we can actually use a instrumentation to measure, or we can see, like the patient tells you they have an itchy rash, and you can see a rash, right? A syndrome is signs and symptoms that characterize a disease, and they often are all clustered together. So you might have a whole bunch of things that you see as signs and things that the patient tell you, and together all of those things are what we expect to see in what we would refer to as a syndrome. Asymptomatic or subclinical means infections that lack symptoms but may still have signs of infection. So, a little bit more here. Symptoms, these are things that are sensed by the patient. Pain, nausea, headache, chills, sore throat, fatigue, or lethargy. Malaise, itching, abdominal cramps, sorry. Those are all things that the patient is telling you that can't really measure. Now, itching, if there's no sign of a rash, is what they mean there. Signs are things that we can actually see. So swelling, redness or rash, vomiting, diarrhea, fever, pus formation, anemia, and it goes on and on. So things that we can actually measure, that would be a sign. So one of the things that really helps in any type of medicine that you are doing is learning some terminology. And so this is a slide that starts the process of learning some terminology. So we always talk about with terminology, when we're looking at a medical term, you have the prefix, which is what's in front of the root of the word, and then the suffix. So they're giving you some examples here along with the meaning and an example. So carcino is the word for cancer. So when we say something is carcinogenic, it means it gives rise to cancer, because genic means like Genesis, the beginning of, right? Or let's look at another one, emia, the fifth one down. Emia is a suffix. So if it's got the little dash in front of it, it's telling you it's a suffix. If it's got the little dash after it, it's telling you it's a prefix. All right. So emia means pertaining to the blood. So viremia means viruses in the blood. All right. So tons and tons of terminology to learn, and a lot of it you just pick up along the way, but there's some that you'll want to try to start learning now. Like itis always means inflammation of something, like meningitis is inflammation of your meninges. The cause of the disease is called the etiology. So the topic of etiology covers the study of the cause of the disease and knowing that diseases have various causes. So when somebody says, what was the causative agent, or what was the etiology? What caused that particular disease? So we look at diseases, and there's different categories for them. So hereditary means that it's genetic, right? Something that you were transmitted from your parents. Congenital means present at birth. Degenerative means results from aging. Nutritional is because they're lacking something in their diet. Endocrine is hormonal. Mental is going to be emotional or psychosomatic. Immunological means it has something to do with their immune system, and neoplastic means an abnormal cell growth. So again, these are terms that you want to know. Flashcards work really good for making sure that you learn all of these. So you want to know the term, and you want to know the description, and then some examples. And then here are some more. Infectious means it's caused by an infectious agent. Iatrogenic means that it's caused by a medical treatment or procedure. Idiopathic means we have no idea. We don't know how this happened. And healthcare associated or nosocomial, it means that it was acquired while you were in the hospital as a little bonus parting gift. When we talk about etiology, the process uses Koch's postulates. And so Koch's postulates attempts to explain what the causative agent of a disease is. So the germ theory of disease sounds logical to you and I because we've been doing it forever now. But remember back in the time when they were coming up with all this was a brand new idea. The germ theory was that, hey, pathogenic organisms are what are causing infections. Again, we talked about this in history at the very, very beginning of this, is that, people attributed all sorts of problems that they had, sicknesses, illnesses that they had, to all sorts of magical things. It could have been bad air. It could have been somebody cast a spell on you, all kinds of things like that. And these guys came along and said, no, there's actually these invisible microbes that you can't see that are causing this. As you can imagine, because they were invisible and can't be seen, people thought they were crazy too. They thought that was just another idea that wasn't going to go anywhere, but it turned out to be true. Robert Koch was the one who developed a set of postulates that we use to prove that a particular pathogen causes a particular disease. So how does Koch's postulates work? So basically how you start out with, you have some agent that you think is making an individual sick, and you're able to get that out of whatever is sick. And that's what they're showing you there. Squeaky the mouse, they're able to pull out some of the bacteria. Let's just say, for sake of argument, that he had a really bad skin lesion, and they were able to pull some pus out of that, and they were able to look underneath the microscope and then plate that up, right, so that they could grow it, right? And so that's the first thing. You have to be able to get whatever it is that's hurting someone or making someone sick or killing them. You have to be able to get it out, and you have to plate it up, and then you have to be able to grow it, and then you take that that you grew in that plate, and you make sure that it's pure. In other words, if there were several different microbes in there, you split it all out so you just have one kind of microbe growing in each plate, and that's what they're showing you there. And then you take what you grew and you put it into a healthy squeaky mouse, right? And then unfortunately that squeaky dies. And then you are able to pull that same microbe out of the squeaky that you introduced it to and examine it underneath of the microscope. That is how you determine this is the causative agent of this. All right. There are, of course, now in the 21st century exceptions to Koch's postulates. We know that some pathogens can't be cultured in the laboratory. Viruses are a perfect example of that. Some diseases are caused by a combination of pathogens and other cofactors, and then there are ethical considerations that prevent applying Koch's postulates to pathogens that require a human host. So we don't do this with humans, obviously. There are difficulties in satisfying Koch's postulates as well, because diseases can be caused by more than one pathogen, so you got to know which one it is that's causing it, and pathogens that are ignored are potential causes of disease. So you think it's one, but then you find out it's actually another. The virulence factors of infectious agents. Pathogenicity is the ability of a microorganism to cause disease. Virulence is the degree of the pathogenicity. In other words, how sick does it make you? Virulence factors contribute to this ability to make an individual sick or not sick. And those include adhesion factors, the production of biofilms, extracellular enzymes, toxins, and antiphagocytic factors. So this is a little chart looking at the relative virulence. How sick will this make me of some microbial pathogens? So it's showing you up at the top, rabbit fever and the plague and whooping cough. So the ones at the top are the most virulent. I unfortunately don't know anything about rabbit fever. I guess I need to look that one up. But the plague I sure know a lot about, and whooping cough, of course. And then Pseudomonas aeruginosa is the infection that people get when they get burns. C. diff, right? So that's antibiotic-induced colitis. Candida albicans, vaginitis, thrush, yeast infections, lactobacilli. They're showing you that there. So the least virulent ones are at the bottom. So let's look at these virulence factors of infectious agents like extracellular enzymes. These are secreted by the pathogen. They dissolve structural chemicals in the body, and they help the pathogen maintain its infection, invade, and avoid body defenses. They are important to the virulence of the pathogen. Mutant species do not secrete the enzymes. These are often called avirulent. So sometimes you have viruses in your body or pathogen in your body that shouldn't be there, but they don't do anything. And so we say they're there, they're present, and we refer to them as being avirulent because they're not able to make you sick. So here it's just showing you how some of the extracellular enzymes work. Toxins, these are chemicals that harm tissues or trigger the host immune response that cause the disease. So the bacteria releases these chemicals called toxins. Toxemia refers to the presence of toxins in the blood. The toxins are carried beyond the site of infection, and there's two different types of toxins that we talk about, exotoxins and endotoxins. So here it's showing you exotoxins that are outside of the body, endotoxins which are inside of the cell itself. So a comparison, I may have said that wrong. Exotoxins are outside of the cells and in the fluid, not outside of the body. And endotoxins. So here they're just showing you some examples of the differences between exotoxins and endotoxins, the source of them, the relationship to the bacteria, the chemical nature, toxicity, and so on. So you can read through that and understand the differences between endotoxins and exotoxins. Antiphagocytic factors, these are factors that prevent the process of phagocytosis by the host's phagocytic cells, so the macrophages and other white blood cells. They allow the pathogens to remain in the host for a longer time because they don't stop them. One of the things that is considered an antiphagocytic factor is a bacterial capsule. So we looked at capsules in lab. You had a capsule stain that you looked at. These are composed of chemicals that are not recognized as foreign, and they are slippery and difficult for phagocytosis to occur. So phagocytic cells simply just can't get a hold of them. And then antiphagocytic chemicals, these prevent fusion of lysosome and phagocytic vesicles. Leukocidins directly destroy phagocytic white blood cells. So here it's showing you phagocytosis being blocked by the capsule on the outside of the bacterium. All right, looking at the stages of infectious disease. So when we talk about diseases, they follow a very typical pattern. So there are usually 5 stages following an infection. There's the incubation period, the prodromal period, the illness period, the decline period, and then convalescence. So taking a look at that and what each of those mean. So incubation is when you don't have any signs or symptoms. It's when they say, oh, what you could be infecting other people, and not even knowing it. The prodromal period is when you don't feel good. You have some vague, you maybe got a slight headache, maybe you feel just a little off or a little feverish or whatever. The illness, obviously something's going on. You have severe signs and symptoms. Decline is when those start to go away. And then convalescence is when you no longer have any of the symptoms or signs of the illness. So, over on the left-hand side of your graph, it shows you the number of microorganisms that you would have. And it's not giving you any number. It's just showing you that obviously the most number you're going to have is when you're in full throes of the illness. And then across the bottom is time. And of course, that's going to be different too. So, if we're talking about a cold, that might be 7 to 10 days. If we're talking about something else, it might be a month. It just depends. So here is a table that just looks at some of the incubation periods of a few selected diseases. Staphylococcus in foodborne infection, in other words, food poisoning. The incubation period is less than a day. So if you've ever had food poisoning, you know that. You go eat something and you wake up in the middle of the night and you're really sick, or you eat something late in the day and then the next morning you don't feel well. It's usually less than a day. Influenza, about a day. Cholera, 2 to 3 days. And you go on down the list. So you can see some of them, like leprosy, the incubation period for that can be upwards of 30 years. So you may have been exposed to it, and you're carrying it around this whole time and you have no idea. And 30 years later, you end up with Hansen's disease or leprosy. And then finally, the movement of pathogens out. So we talked about the portal of entry. Now we're going to talk about the portal of exit. How do the pathogens get out? So they have to leave the host through a portal of exit, and many of the portals of exit are the same as the ones we saw for the portals of entry. So pathogens often leave the host in materials that the body secretes or excretes. So looking at portals of exit, again, some of them are the same. Earwax, tears, nose secretions, saliva, those kinds of things. So there's a few that were not considered portals of entry that are considered to be portals of exit. Let's talk a little bit about the modes of infectious disease transmission. So transmission is from a reservoir or a portal of exit to another host's portal of entry. And so there's three groups of transmission that we talk about: contact transmission, vehicle transmission, not your car, and a vector transmission. Contact transmission is direct contact. You can have direct contact transmission, means that that involves body contact between hosts. So transmission with a single individual can also occur. Indirect contact transmission is when pathogens are spread from a host to another host by fomites, and fomites are inanimate objects. Like, I always think about the playgrounds that they have at some McDonald's that have those big pit of balls, and those balls are all fomites because they're covered in pathogens. And one kid goes in and he picks up the pathogens from another kid. He didn't have any contact with the other kid, but he did have contact with something the other kid had contact with himself. And then droplet transmission. This is the spread of pathogens in droplets of mucus by exhaling, coughing, or sneezing. One of the things that I continuously told people back during COVID is that anytime you've had a respiratory infection, cold, virus, COVID, whatever it was, and it was classified as a respiratory infection, you got that because you inhaled someone else's nasal secretions or oral secretions. In other words, you inhaled someone else's snot or someone else's spit. So think about that for just a minute. That's why you want to wear a mask around people that are sick. And when you're sick, why you want to wear a mask. So please wear a mask when you're not feeling well so that you don't make other people ill. They're showing you droplet transmission. I've seen some better pictures of this than the one that your book supplies, but just a single cough or sneeze. I don't remember what the estimate is, but it can be like 10 feet. So somebody can be hacking and coughing in the other room, and you walk through it and you inhale that droplet transmission, and now you are infected. Vehicle transmission is a couple of different ways. This is using either air or water to move the microbes. So in airborne transmission, the pathogen can travel, here's an idea about it, about a meter or more via what's called an aerosol. So aerosols can occur when you sneeze, cough. They can also occur through air conditioning systems and through sweeping the floor. So hantavirus out in New Mexico in the desert Southwest was originally being transmitted because people it comes from rodent feces, and when the rodent feces would dry, and people would sweep that out of their house, it would create an aerosol and they would inhale it during sweeping. And then waterborne transmission. This is important in the spread of a lot of gastrointestinal diseases, bunches and bunches and bunches. That's why we're so careful about our municipal water. And if something has happened to the municipal water supply, they're real quick to put out boil water alerts. I know we get them a lot down here in Savannah, especially after we've had a big hurricane event or even tropical storm event, because we get little breaks in our water system. Our water main system is very old down here, and there's always a possibility that it can start spreading gastrointestinal diseases. Another term that's used for waterborne transmission is the fecal to oral route. In other words, there's fecal material in the water and you ingest it. Foodborne transmission is the spread of pathogens in or on the food, so inadequately processed, cooked, or processed, cooked, or refrigerated foods. Sometimes food can be contaminated with fecal material from another human or from rodents or all kinds of lovely thoughts. I hope nobody's getting ready to go to lunch or dinner because this is probably pretty unappetizing. Um, Bodily fluid transmission. This is the one you have to really be concerned about in healthcare, like blood, urine, saliva. They can all carry pathogens. You want to prevent contact with conjunctiva, which is the lining of the eye, or breaks in the skin or mucous membranes. You want to make sure that none of those types of fluids come in contact with those areas. So poorly stored foods can harbor pathogens and transmit diseases. Um, many years ago, I had the privilege to live in another country, and unfortunately, it was not a first world country. It was a third world country, and I was surprised at how often I got food poisoning. And part of it was because of the lack of refrigeration and the lack of things being stored properly and tested and all that sort of thing. And this is very typical of what you see in some countries where in the United States, you never see things like this. Everything's kept refrigerated because we know that refrigeration keeps down the transmission of pathogens and the development of pathogens, the multiplication of pathogens, all that sort of stuff. But in other parts of the world, that's not exactly the way it is. So when I lived overseas, I very quickly became a vegetarian because it seemed like every time I ate some type of meat, chicken, anything like that, I got sick. So I learned in a hurry that the best thing to do was to stay away from all that stuff. Vector transmission, there are two kinds of vector transmission we talk about here. There's biological vectors. These transmit the pathogens and serve as host for some stage of the pathogen's life cycle. Biting arthropods, insects, transmit many diseases to humans. So they're biological vectors. Mechanical vectors are passively transmitting pathogens because they're present on their body. And so flies are examples of mechanical vectors. They land on things out there that you don't want them to land on, and then they go land on your food, right? And so they are mechanically passing the pathogen over to you. Here's some selected arthropod vectors. The ones we always think about are mosquitoes. Mosquitoes are one of the absolute worst animal vectors there are. So mosquitoes are animals, insects are animals, and there are lots of different species of mosquitoes. The one that a lot of people think about is the Anopheles mosquito, which is the one that causes malaria. We don't see a lot of malaria in the United States anymore, thank goodness, but we used to. But there's other mosquitoes like the Aedes and different species of Aedes that cause yellow fever and elephantiasis and dengue fever, and viral encephalitis. So you can see that there's different species that are causing this. Another biological vector that a lot of you are familiar with are ticks. And so they give you some of the different types of ticks and the diseases that they cause. Fleas, lice, all those kinds of things all cause different diseases. Blood-sucking flies are another example of a biological vector, and they're giving you some example of those, as well as blood-sucking bugs. Um, just recently, in the last year or two, um, I had always taught about Chavez disease, which is a disease that you would see in South America by a bug called the kissing bug, because it would come out of thatched roofs and because that's where it likes to live in the night, and it would bite individuals near their mouth. That's why they called it the kissing bug, and it would create this disease called Chavez disease. And we didn't see any in the United States, but I had been reading in the last year that there is an increased incidence of Chavez disease in Florida. So it's on its way into the United States for sure. Mites or chiggers, you might have gotten chiggers if you went to day camp or you went camping or anything like that. They can actually cause a disease. A specific type of them can cause a disease called scrub typhus. Some mechanical vectors, I mentioned flies before, house flies. They land on unsavory type things and then they go land on your food. Cockroaches, same kind of thing. They crawl through and climb on cockroaches, get on anything, and then they climb in your food. So all of them are going to cause different types of foodborne infections, including Shigella and Salmonella and E. coli. Contact transmission, just a little chart for that. Direct contact is things like handshaking, kissing, sexual intercourse, bites. So this is a real big problem with animals biting people and with kids biting each other. You see that in daycare sometimes. And then examples of some diseases that can be spread by direct contact. Indirect contact is when you have things like you sharing something, like drinking glasses or toothbrushes or toys or anything like that. Punctures like needles, right? Um, and then droplet transmission, sneezing, coughing, and all that sort of stuff, and then diseases. Vehicle transmission, airborne, waterborne, and foodborne, and it's talking about each one of those and some examples of different diseases that can be transmitted that way. And then finally, vector transmission, either mechanical or biological. So a nice summary chart on the modes of diseases so that you can kind of organize that for yourself. Let's talk about classification of infectious diseases. So we can classify diseases a couple of different ways. Taxonomic categories, the body systems that they affect, their longevity and severity, how they spread to their host, and the effect that they have on populations. So this is the kind of stuff that epidemiologists do. This is what they really get into, is they're looking at statistics and they're looking at all of this kind of information that we'll be talking about. So again, a lot of terms in this chapter. So some terms that you want to be familiar with. Acute disease is a disease where the symptoms develop really quickly and then they run its course pretty quickly, like a cold, like a common cold. Chronic disease is when they have mild symptoms that develop slowly and last a long time. A subacute disease is one that there is a range of time course and symptoms between the acute and the chronic phase. Asymptomatic disease, this means you obviously don't have any symptoms. Latent diseases are ones that appear after a long time in incubation. Remember, we were talking about uh, Hansen's disease a few minutes ago. It could show up 30 years later. Communicable diseases are ones that are transmitted from one host to another. Contagious are ones that are easily spread. Non-communicable diseases are ones that are not passed from person to person. Local infection means it's limited to a small area of the body. Systemic infection means it's widespread. It's everywhere in the body. It travels through the blood and the lymph. Focal infection means that the infection site has served as a source of pathogens for infections to other parts of the body. And primary infection is the initial infection within a given patient. A secondary infection is what develops after the primary infection. A classic example of a secondary infection is you have a really bad cold and you end up with a sinus infection, right? So you had a viral infection that was not really a big deal, but unfortunately it trapped some bacteria in your sinuses and you got a sinus infection. The sinus infection would have been the secondary infection. Some other things that we talk about in epidemiology, the frequency of disease. So disease occurrence is tracked using two separate measures. Incidence is the number of new cases of a disease in a given area during a given time period. So it's the number of new cases in the county you live in, or in the part of the state you live in, or in the entire state, or in the entire United States, or the entire world. So the parameters are given for the incidence. Prevalence is the number of total cases of a disease in a given area during a given time period. So incidence is new diseases, prevalence is total. The occurrence also is evaluated in terms of frequency and geographic distribution, and that's what I was just talking about. We might be talking about the number of new cases in just your college, for example, or in your dorm even. Or we may be talking about it in the entire city that you live in, or the region of the state that you live in. So again, it's going to evaluate depending on how they're looking at it. So looking at this curve represents the incidence and estimated prevalence of HIV/AIDS among U.S. adults. And so on the left-hand side, you're looking at the incidence in thousands, and across the bottom, you're looking at the year. And so the incidence is red and the prevalence is blue. And again, just what I was just saying, epidemiologists report data in a variety of different ways. So there's lots of different statistics that they use. So if you like statistics and you like graphs, epidemiology is a great career because you're always explaining data. So some other terms that we want to know. So endemic is the first one there, meaning that it's everywhere, sorry about my cat, everywhere in low amounts, but it's persistent all the time, but in small amounts. Sporadic means a little bit pops up here, a little bit pops up there, but there's no true pattern. An epidemic is when we have a high incidence of it in a particular area. So you see the red dots there in South America. And then pandemic, which we all unfortunately know way too well, means that it's everywhere. Epidemics may have fewer cases than non-epidemics, and so they're showing you some graphical material here. We think about epidemics as having huge numbers of cases, and that's not always the case. Sometimes, because epidemics are concentrated in one particular area, so you may have a concentration of one area, but there may be fewer of those when we spread that out over a larger area. All right, there is a list of nationally notifiable infectious diseases that if a case of this presents itself in your practice, you have to notify the CDC with regards to that. In some cases, you have to notify the state and the CDC. And so every state has a different list for state notifiable, but nationally notifiable. And you'll see all of these. I by no means expect for you to memorize this. This is something that changes from time to time anyway, but you simply can look it up, and a veterinarian or a physician in your office would know which ones of these diseases they have to report. So here's the first page of them, and the second page, the third page, our fourth page, and finally the fifth pages. So this is where hospitals, physicians, and other healthcare workers are required to report cases to their state health department and then forward that data to the CDC. So again, do not memorize that list. Just realize that there are lists out there that you would have to pay attention to, because some of these are animal zoonotic diseases. So they are diseases you would need to be aware of if you have cases of them. And here's just showing you more statistics. Like I said, if you like statistics, man, epidemiology is your thing. This is the Nationally Notifiable Infectious Disease Weekly Tables. You can actually access this by going to the CDC website. So I don't know how much of it is currently available, but you can look this information up and it'll give you an idea of what's going on in different parts of the United States with regards to different diseases. Epidemiological studies. Descriptive epidemiology uses careful tabulation of data concerning the disease. It records the location and time of the cases of the disease and collects patient information. It doesn't collect their name or their social security number or any personal data like that. It collects information like how old they are and what gender are they, and all that kind of information. And that allows them to try to identify what's called the index case of a disease, which is where did it start from. So there's a classic story that we always talk about. This is what they were looking at when they were trying to figure out the index case for cholera in London in 1854. And so there's a fascinating book called The Broad Street Pump, that talks about how all of this was done. So, again, if you're interested in epidemiology, it's a great book to read. And it talks about how they actually went through the process of determining where the source was for this cholera. It was coming from a water pump on the Broad Street in London. So during 1854, they didn't have indoor plumbing, so you had to take your bucket every day and go down to the neighborhood pump, and you had to pump the water. Well, they also didn't have sewer systems, and so unfortunately, people tossed their waste from chamber pots from inside their house into an area, and that was seeping down into the water supply, and then they were pumping the water back up. And so they ended up with massive cases of cholera and a lot of people dying. And so this red dots right there are just clusters of people that were dying from cholera, and they were trying to trace where is this coming from. And so they went through this whole process of figuring that. It's a really fascinating story about how they figured this out, considering they didn't have they didn't have an internet or anything like that to do this with, so they had to do all of this manually. Analytical epidemiology seeks to determine the probable cause, the mode of transmission, and the methods of prevention. It's useful in situations where Koch's postulates can't be applied. It's often retrospective, meaning we look back after something has happened and do a lot of thinking about what happened. There is a series that was produced in 2019, I believe, or 2018. Maybe it was 2018, and it was on the Black Death, and it was done by a professor. I can't remember where she's from now, but it was done by a professor, and it was all retrospective, and they were able to use a lot of data that they hadn't ever all put together on the Black Death before, and they were able. Purdue, this professor went to Purdue. Sorry, things come to me suddenly sometimes. And it was a really interesting series. The company used to be called Wondurium. I think it's got another name now, but you could subscribe to it, and sadly I did during COVID when the lockdowns were happening, and it was just fascinating because it was a multi-part series about the Black Death and what they now know based on all of the science that we can do today. So this is what they mean by analytical epidemiology. They're looking back at something that happened 500 years ago, more than 500 years ago, and making determinations based on the current ways that we're able to look at things. Experimental epidemiology is testing a hypothesis concerning the cause of the disease. So this is typically current. So this is the application of Koch's postulates, and it's a lot of what you see in emerging illnesses, developing and emergency illnesses and diseases. They're applying Koch's postulates, trying to figure out what it is that's causing it. Hospital epidemiology, or something we all have to be aware of if you work in a healthcare environment, especially if you're a nurse, but it's important for patients as well. So healthcare associated or nosocomial infections, there are several different types of these that can happen. There's exogenous, meaning the pathogen was acquired from a healthcare environment. Endogenous means the pathogen arose from the normal microbiota within the patient. So you had some type of a mutation occur. Iatrogenic means it results from a modern medical procedure, and superinfections, which is the use of antimicrobial drugs that inhibit some resident microbiota, allowing other microbes to thrive. So there's always a complicated interplay about how healthcare associated infections happen. So hospitals and healthcare facilities have microbes in them because there's people there and there's sick people there, and you've got immunocompromised patients there, and then you have the transmission of those pathogens between the staff and the patients and between the patients and all kinds of things, and it kind of all comes together in the perfect storm. There's been a lot of interesting research that's been done about nosocomial infections and about how they're transmitted. Every hospital has an infection control nurse, and that is the person who is responsible for going around basically swabbing everybody's hands and nasal cavities to make sure that you're not passing infection on to patients. But one of the interesting research practices or research experiments that they did in Australia was they were trying to figure out how this particular infection was passing from individual to individual in a hospital. And what they found out was that it was being passed on a physician's tie. So physicians used to go visit patients first thing in the morning, and they usually were wearing a suit and tie when they did that. It was considered proper to do that. Now you see them in scrubs. But those ties, how often do you wash or launder or dry clean a tie? The answer is almost never. And so if you're in a hospital setting and you've got patients that are coughing up on your tie all the time, and then you're going to care for another patient, same issue, right? You're passing things from one patient to another. And so that's one of the reasons why you see physicians rarely in street clothes. You almost always see them in scrubs. And the same with everyone in the hospital. Most of the time in hospitals, at least where I am, I don't know how it is in other parts of the country, but you don't get to leave in those scrubs at the end of the day. You come in your regular clothes, you change into your scrubs, and then you leave the scrubs there, and then you go about your business. And it doesn't matter if you're a patient care transmit person or you work in in dietary or whatever it is you do. You don't wear your clothes that you wore in the hospital out, and you don't wear what you wore into the hospital because of this risk of passing diseases. How do we control these? It requires aggressive control measures. So things like I was just talking about, controlling clothing that's leaving and coming and going, and shoe covers, and all those kinds of things. Hand washing is the big one. It is the most effective way to reduce healthcare associated infections. I know people like to use the gels and the antibacterial, all that sort of stuff. I know people like to use it because it's quick and it's fast, but really and truly, I don't remember what the current recommendations are now, but it used to be you could use those 3 times and then you had to do proper hand washing. So I don't know the current recommendations, but you don't want to rely just on sanitizers, right? You want to actually wash your hands. That is the best way to prevent passing infections to one another in a hospital, to a patient in a hospital setting. It's also the best way to prevent in your own home. So washing your hands, when I go outside and I come back inside, I live on a farm. And so when I go outside and I see my goats, and I feed my goats or I see my chickens or whatever it is, I immediately come in. Number one, I don't wear the shoes that I wore out there in my house. And number two, I wash my hands. Wash my hands immediately. Because even if all I did was pet a goat, I could potentially be bringing pathogens into my home. What about epidemiology and public health? So sharing of data among public health organizations is critical. It is important that we're keeping all of this data because it allows people that look at this, public health officials that look at this, to try to manage disease. So there are agencies at the local, the state, the national, and global levels. The U.S. Public Health Service includes the National Public Health Agency. The CDC is one branch of that. And of course, the World Health Organization, which coordinates public health efforts worldwide. The role of public health agencies in interrupting disease transmission: they work to limit that disease transmission. They enforce cleanliness in water and food supplies. That's critically important in certain parts of the world. We are very fortunate here that our water is usually safe, and that our food supply is typically safe, and that we have a methodology for when we think the food or water is not safe to alert the public. Um, they work to reduce disease vectors and reservoirs. There's been a lot of work in recent years, making sure that individuals in certain parts of the world are able to filter water so that they're not they're not infecting themselves with pathogens that are in water. They establish and enforce immunization schedules. They locate and treat individuals that are exposed to contagious pathogens, and they establish isolation and quarantine measures when someone is ill and we want to make sure that it doesn't start to spread. A little bit about public health education. Diseases that are transmitted sexually and through the air are difficult to control, and public health agencies campaign to educate the public on healthful choices to limit diseases. All right, so lots of definitions in this chapter. And so I would really pay attention to definitions. And when you have sequential events like the states of a disease, where you have the incubation period and all of those kind of things, always take a look at those kinds of things. The charts are helpful in looking at those things as well. So once you have listened to the lecture and you have done your homework in Pearson on this chapter and studied a little bit, you should be ready to take your quiz. And I will see you for the next chapter. Bye.